Okay, are we good to start?
We've got the attendees are coming in.
Great.
We might just wait a couple of minutes.
Yep.
All right, you're good whenever you're ready, Julie. Thank you.
Okay, welcome everybody to Australian Wildlife's Week, Australian Wildlife Society Wildlife Week webinar. Just to start off, I'd like to do an acknowledgement of country. So I'd like to begin by acknowledging the traditional custodians of the land on which we meet today and to pay my respects to elders past and present. I would like to extend that respect to Aboriginal and Torres Strait Islander peoples here today. So it's Australian Wildlife Week. Welcome. My name is Dr. Julie Auld, and I'm the president of the Australian Wildlife Society. Australian Wildlife Week is about commemorating across the country the positive relationship between humanity and nature. The Society started or founded Australian Wildlife Week in 2019 because we wanted to make people more aware of our environment and wildlife conservation issues. We want to explore and develop a deeper understanding of these issues, gain the necessary skills to make informed decisions, and implement wildlife conservation action. One of the issues we're currently dealing with is the amount of plastic in the environment. And hopefully you're all well aware that we have open a petition called Snip Rings for Wildlife, and it closes tomorrow. We'd really like to make our target. So if you have not already signed that petition, we'd ask that you go in and participate with that and sign our petition. So the webinar, how's this going to work today? Okay, well, we're going to be showcasing all the wildlife conservation and research conservations across Australia. And we've got 5 keynote speakers today, as well as 6 students that have been awarded our Australian Wildlife Society University Research Grants. So they're going to be talking about their research. And to be honest, I'm really excited about today. Few housekeeping things. Your video and audio will be muted, and each speaker is going to be introduced by one of our directors. If you have a question, you can pop it in the question and answer box. You'll find that section down the bottom of your Zoom screen. And obviously, I can see a few people have started to use the chat. Feel free to use that chat as well. When the questions start, our National Office Manager, Megan, will be asking the key speakers for their response. So she'll work through those question and answers on the question and answer window. So this webinar is being screened live on Facebook, and we're also recording it, and it'll be available on our website, hopefully soon afterwards for people that were unable to attend. So if you have any general little questions, maybe put them in the chat. And if you have questions and answers for the keynote speakers or our student presentations, please put them in the question and answer box. So we're off and running with our first speaker this morning, and I get to introduce Kyle Brewer. So Kyle is a student from the University of South Australia, and he's going to be speaking about pH responsive 1080 plants for the mitigation of catastrophic predation of native animal populations. Thank you, Kyle.
Thank you for that, Julie. I'll just quickly share my screen. All right. So as Julie said, I'm going to be briefly discussing some of my PhD research. And we've got a little bit of time, so I'll just get straight into it. So judging by the audience at AWS, I think most people would probably know how problematic feral cats are for Australian mammals. Essentially, they are the single greatest predatory threat to native Australian mammals. I'm going to quickly highlight two of the reasons why that is and how that eventually relates to the context of my work. So to start off with, feral cats are basically everywhere. They cover almost the entirety of the country, and there are millions of them, and they're only absent from a couple of secluded island refugia and wildlife sanctuaries where they're actively controlled. And in addition to that, they also kill, on average, over 800 million native mammals every year. And the overwhelming majority of these are native species, and that amounts to quite a significant loss of biodiversity and negative impacts for our native mammals in Australia. And one of the main ways in which we try to undo some of this damage is to reintroduce threatened species through reintroduction programs. And there's a particular problem associated with that, and that's really what my research focuses on. So if we imagine a typical reintroduction program, generally we start off with our reintroduced population, and before we reintroduce them to a target area, we make attempts to remove all the feral cats from within that area. And there are a bunch of conventional control methods that we use, like poison baiting, trapping, etc. But all of these have their own particular shortfalls. And generally, it's quite challenging to remove all the feral cats from an area. And in addition to that, it's difficult to prevent feral cats from invading that reintroduction area during the program. And generally, that leads to the persistence of feral cats that are either resistant to control methods or simply invade that reintroduction area. And generally, we call those kinds of feral cats problem individuals because they're responsible for the majority of the depredation of native mammals in reintroduction programs. And in many cases in the literature, we see that a single problem individual can be responsible for essentially preying upon all of the native mammals in a reintroduction program. And within a few short weeks or months, they're all wiped out, and that results in the failure of the program, which highlights the need for us to be able to control specifically and these problem individuals. And that's where my research comes in. So the solution that we've proposed to address this predation was actually proposed by an entomologist by the name of David Peacock about 25 years ago. And I'll briefly just go over the concept. So essentially we have our reintroduced population or native mammals that are to be reintroduced. And we proposed that if we were to have some sort of poison implant that had a pH responsive coating, we could subcutaneously implant that into the entirety of the reintroduced population. Keeps this encapsulated poison stable at physiological pH. But in the event that a problem individual preys upon one of the animals in that population, it ingests that poison implant. And due to the low pH gastric environment of the feral cat or the problem individual, that pH responsive coating is dissolved, releases the encapsulated poison, and kills the problem individual, thereby leading to the protection of the remaining native mammal population, which is where we get this population protecting implant name from. So in my PhD, I've been focusing on developing and optimizing the manufacture of the implants, as well as doing some preliminary testing of them. So to manufacture the implants in the lab, we start off with this instrument here, which is a fluidized bed spray coater, a piece of equipment that's commonly used in the pharmaceutical industry. I won't focus too much on how it works, but essentially we start off with a small piece of ABS filament, which is cylindrical in shape, just under 8 millimeters in length and just under 0.5 millimeters in diameter. We then coat on top of that what we call a core formulation, which could contain sodium fluoracetate or Canadian, and we can coat a lethal dose of 3 milligrams on that, in addition to a binder and a dye, which helps to form a cohesive coating on the piece of plastic. And for the majority of my development, I actually use a compound called sodium acetate, which is a non-lethal analog to sodium fluoracetate, to allow me to do some lab scale development and manufacture. On top of that, we then coat a pH responsive formulation, which enables me to tune the coating thickness, which we can investigate its effect on the operation of the implant, the performance of the implant. And just to note, this lab scale approach enables us to make roughly 1,000 implants in the 1 to 2 week turnaround. And just to quickly cover some results, here we have implants that have been prepared at the top, just an implant final product. And then below that, we have a cross section of the implant where you can see the white ABS filament scaffold that we use, the purple core of the implant, which contains sodium acetate in this case, but could contain 1080, and a clear coating that is our pH responsive coating. And here a bit of reference to size, we can see a standard RFID microchip at the top on that zoomed in insert, and on the left, a standard conventional syringe microchip with syringe implant that's used to implant RFID microchips. And at the bottom, we have one of our population second implants, and you can see that it's quite a bit smaller and it can easily be incorporated into use of a syringe implanter, which means it could be used in the field quite readily. And just to quickly give a little bit of points about its performance, at pH 1.5, which is a stimulated gastric environment, we can see that if we increase the coating thickness of the pH responsive polymer, we can control the percentage release of the dye that's in the core as a proxy for poison release. And overall, we can see that even at the thickest coating, we have release occurring within a couple of hours, which is well within the time necessary to have it released in the stomach of a feral cat. We've also performed some preliminary in vivo trials where we've demonstrated that the implants are stable within the subcutaneous environment of rats for 12 weeks. So going forward, I've already managed to prepare some implants that contain 1080 and haven't seen a change in performance in vitro. But our next steps are to look at the in vivo stability of the implants, as well as support them in field trials. I'd just quickly like to thank my research group, my supervisor and co-supervisor, as well as our collaborators that have helped us develop the population of implants, and in addition to our funding sources and AWS. Thank you.
Thank you, Kyle. We're going to go to the next speaker now, and Patrick is going to introduce the next speaker.
I just need to unmute Patrick. Give me a second. All right, Patrick, can you please unmute?
Unmute. Yeah, good morning and thank you for that. It's my real pleasure. I'm Patrick Medway from the Australian Wildlife Society. It's my delight to introduce our next guest speaker and our first keynote speaker, Jamie Trezesi, from the reclaimed Kosciuszko. He's going to speak about the impact of feral horses on the Snowy Mountains, the Kosciuszko National Park. This has been a real problem for us for a long time, and we're delighted now to have Jamie explain what's happening and how it's going to be made much better for our natural environment in the Great Snowy Mountains. Thank you, James.
Thanks, Patrick. Yes, and thanks to AWS for having us. It's a great privilege to be able to talk to you all. I'd like to start off by acknowledging that I'm presenting from Ngunnawal land, and I wish to pay my respects to their elders past, present and emerging, and to any First Nations people on the webinar today. I will attempt to share my screen and see if this can work. There we go. Hopefully everyone can see that okay, and you can't see my talking notes.
Yes, looks good.
Great. So my name's James Trezesi. I'm the Conservation Director at the Invasive Species Council, but as Patrick also pointed out, closely involved with the campaign at Reclaim Kosci. And I don't know if anyone's noticed recently, but there has been a pretty seismic shift in both the politics of New South Wales, but also what is happening around feral horses in Kosciuszko over the past 3 days. So I was literally rewriting this entire presentation last night, just based on the news that happened on Sunday. So it's a very mobile and nimble space, but I'd welcome to take any questions from those people in the audience. Kosciuszko is one of the jewels in our national parks state. it was established as a state park in 1944 and then evolved into a national park in the 60s. It is the largest national park in New South Wales, and it has ecosystems and wildlife that occur nowhere else in the world. It is a really important place. It is also a place that has a huge variety of impacts. So both human-driven impacts, such as the development of ski fields, snowy hydro. There's a huge controversy with more recent developments around special activation precincts, but also human indirect impacts, I would argue. So feral horses being key to that, but also feral pigs, feral deer, a whole range of invasive species, orange hawkweed, etc., that dramatically affect that landscape. And so when we talk about what is in Kosciuszko, why it matters so much, why all this effort to try and control feral horses, well, we often in the media and in public commentary, the conversation always ends up being about the horses, and we lose sight of what we're actually trying to do. I don't wake up every day going, how can I take out horses? I wake up every day going, how can I help protect the landscapes and wildlife of Kosciuszko? That's what motivates me. I've nothing against horses. I think they're a lovely animal in a paddock where they belong. But in sensitive alpine and subalpine ecosystems, they do huge amounts of damage, and that is what we've learned. So on your screen are some of the incredible species that occur in Kosciuszko that are found nowhere else in the world. Northern corroboree frog there, but it's also got its southern cousin. Alpine sheoak skink, broad-toothed rat, and pretty sure that is an anemone buttercup. So there are more than 50 nationally threatened species that occur in Kosciuszko, and there are 34 threatened species listed under national and state law that are directly threatened by feral horses in that landscape. Kosciuszko also is the headwaters of 3 of our most important inland rivers. So the Murrumbidgee, the Murray and the Snowy. All their water starts in the alpine landscapes and subalpine landscapes of Kosciuszko National Park. So when we look at why feral horses and the importance of taking action, we need to understand the damage that they do. And it is serious and significant and getting much, much worse, exponentially worse, as I'll show you shortly. They trample and degrade water-retaining peat wetlands and sphagnum bogs, which are a critically endangered ecosystem in their own right. But these are giant sponges that have evolved over millions of years to hold water, hold snowmelt, and release it gradually over the warmer months. And so they are like a reservoir, nature's own dam, so to speak, that will slowly release water in the drier months and support many different types of ecosystems and communities downstream. Now, horses do what you can see here. That's a sphagnum bog, which on the left-hand side, that has been seriously damaged by feral horses, and you can actually see the hoof prints in the mud. On the right-hand side, that's the Tatangra wetland. That's just near Tatangra and the wetlands there, and you can see what they're doing to that kind of, nice peatland bog system. And that's Corango Creek. And you can see they're the type of damage horses do. They also overgraze and degrade habitat structure for species like the broad-toothed rat, which require tussock ecosystems to be intact. And so you can see here, that's an exclusion plot where on one side the horses have been excluded, and on the other side you can see the kind of damage that horses do. And that is right near, that's in Co-Wombat Flat, right near the headwaters of our major river systems. So we often talk about, the iconic, or we often hear from the media and from others about the iconic nature of horses, but we don't really see this imagery in the public discourse. We don't talk about what species we're trying to save and also the types of damage that are getting done. And the big challenge with that is horses are a big, large mammal. And so that's what people like to focus on. So one of the big challenges with the debate around feral horses in Kosciuszko is how polarizing it is. It is a very divisive issue, and that comes with a very serious cost. But one of the challenges that we have in kind of communicating the scientific damage or the damage that horses do to the Alps and their ecosystems is cutting through, I guess, those the same arguments that we see from kind of the anti-vaccination and climate denial sectors, which is they surround themselves in a social media echo chamber. They will deride any science that disagrees with their point of view, and they'll largely become this self-fulfilling entity that will attack anyone that disagrees with it. So the capacity for rational negotiation or debate around this really diminishes rapidly. And that is a problem, not just isolated to this issue, and we can see it playing out in lots of other issues in our society, but it is something to be acutely aware of, particularly if you're going to stray into the social media debate on this issue, because we on the other, as the people who see the back end of the Facebook comments, it is pretty ferocious and vicious, but it also needs a lot of people standing up and speaking out for our native wildlife. It's one of the craziest campaigns that I've ever worked on. So where are we at now? Well, I talked about exponential growth, and that's what's happened with feral horses in Kosciuszko. So we've gone from 3,000 horses in around 2000 and to about 14,000 horses plus now. And that's probably more around 16,000 because that data is already about a year or two old, and the horse population grows at about 18% per year. So that problem, those photos that I showed you, that is just getting worse and worse. The damage that horses are doing in that landscape is getting worse and worse. Why is it getting worse? Because the management of feral horses in Kosciuszko has been entirely hamstrung by those same political forces that I was just talking about earlier. That control methods to reduce the impacts of feral horses have been entirely stymied. And so we had a plan in 2016 to try and tackle feral horses, and that got rolled. And then we had Australia's first piece of legislation that prioritises the protection of an invasive species introduced into the New South Wales Parliament and passed, that says actually the horses trump the natural environment in Kosciuszko. And so that has been a real disaster for the protection and management of feral horses. And as you can imagine, over the weekend, the chief proponent of that legislation has resigned. And so the political equation around this has shifted quite dramatically. When we talk about management control, we are talking about lethal control. And when we're talking about 14,000 horses, it is one of the key measures that we need to be able to drive down the number of horses in the park. Now, ideally, we wouldn't need to use lethal control. We could use trapping and rehoming, which is where horses go into salt lick pens or attracted to molasses and are then taken off to people who want to rehome them. Now, it's kind of a cutting off your nose to spite your face exercise that some Brumby advocates have gone through here, because by stopping effective control of feral horses, they've made the problem far worse. And by making the problem far worse, to get the problem under control is going to involve, a larger number of horses being taken out of the park. That's the reality. That is the simple reality that by not taking action, we're just compounding the problem, and it's in the data and it's on the graph that you can see right there. We're not opposed to trapping and rehoming horses. As I said at the beginning of this, I don't wake up every morning saying, how can I take out a horse? I wake up every morning saying, how can we save Australian wildlife? So that's the challenge that we have now. And we have a new draft plan that was released on Friday. Actually, it wasn't a draft plan from Kosciuszko. We've been waiting on this for 3 years. It's 100 days overdue from when it was originally meant to come out. It was meant to come out in 2018 and then, 2019, then delayed to 2020 and the bushfires, and it's been continually delayed. But we've got 4 different reports. And I'll try and just give you the crib notes on it because I appreciate we're running out of time. But what the draft plan recommends is a reduction from 14,000 horses to 3,000 horses. Now, that reduction is really important. I'm not going to shy away from the fact that reducing the current number of horses is really important, but there are a few downsides in what the draft plan has proposed and what has evolved out of the Wild Horse Protection Act for Kosciuszko. And you can see here that 32%, or basically 1/3 of the park, and that's those yellow areas, is going to be retained for horse retention areas. Now, those are largely areas, and you can see the areas of blue, those are largely areas where horses currently exist. The challenge there is that they overlap with some pretty important environmental assets. So in the north, we've got stocky galaxid habitat, we've got broad-toothed rat habitat. In the south, I think there is definitely broad-toothed rat in that Beardbow wilderness area. One of the positives, though, is we're looking to get rid of horses from the western flank of the park, although that does abut state forests, from the alpine regions around the ski resorts and main range, and trying to keep them out of the Jagungal wilderness, which is that middle part of the park. So it is quite a bit of a mixed bag, but we're going to see this new horse number of 3,000 concentrated into those 3 yellow areas. And particularly in the south, there's a problem there with the Victorian border, as you can see, and there's going to be a strong exchange of horse populations going on with Victoria, and it's going to create an ongoing management challenge there. Importantly, for the new draft management plan, it introduces lethal control from ground shooting, but it rules out aerial control, aerial shooting of horses. And that's a problem in that rugged southern area where aerial control is probably going to be the most effective and efficient means to remove horses from the landscape. So it's going to be a pretty wild ride. We're still processing everything. As you can see, there was 4 reports. I spent most of my weekend reading all of that, and then also processing what is going on in New South Wales politics. But it's going to be really critical that anyone who cares about Australian wildlife, that cares about Kosciuszko National Park and the environments that are in, makes a comment on the draft plan. It's probably the most powerful thing you can do at this point in time to try and drive an effective outcome for Kosciuszko. Noting that we're also going to have a by-election in that region, and this issue is going to be quite high up on the issues that will be discussed during that by-election. Reclaim Kosci, and we'll be producing a submission guide and to help organisations and individuals, and a way to keep track of what's going on is just to sign up to the website that we have there, and you can just copy and paste, or sorry, you can't copy and paste because I'm sharing my screen, but you can see the link and just jot it down, or even just visit the Reclaim Kosci website. The other way is to follow us on Facebook, and we'll be using social media to try and keep up the update, as long as we can kind of keep the trolls quiet that pop up quite regularly on our pages. So happy to take any questions. So, and thanks very much for having me.
Stop your share screen. And thank you so much for your talk, James. Very informative, and yes, I would encourage everyone to go on and make a submission to the new plan that's been released. We do have one quick question. We've got 1 minute until the next speaker. So specifically around looking at other methods as opposed to shooting, could you please elaborate maybe on the implications of sterilization of horses? I would say that this could probably still lead to habitat degradation. Would you like to speak more to that?
Yeah, sure. So, well, it's pretty straightforward. We've got 14,000 horses, well, probably more like 16,000 now, but 14,000 horses in the park, and the average lifespan of a horse is 14 years. So sterilization actually doesn't remove horses from the park, it stops them reproducing. So we will be able to tackle the growth rate, but we'll still have 14,000 horses trampling the park for the next decade. And so there is a longer term challenge. There is also just a general kind of consensus out there that sterilization at a population this large and this dispersed won't work, and because you won't be able to get all the mares and be able to tackle that. But I think at the end of the day, we need all the tools in the toolkit. So in saying that, sterilization may become an important part down the track. But the first challenge and primary challenge we have for the population inside Kosciuszko, and this is where I was going at the nose despite your face comment, is that we've let this problem get so out of control that it needs pretty drastic action now, and to a point where we can use trapping and rehoming, sterilization, other toolkits to effectively control horses. One of the things that I think is a missed opportunity in the draft horse plan, for example, is an off-park horse reserve. You know, there is definitely a capacity there to create an off-park horse reserve and move significant numbers of populations outside of what is meant to be a conservation area.
All right, excellent, thank you. Just before we move on, I just wanted to point out that we are getting a lot of questions via the chat and the Q&A box. So just letting you know, we will be, if we don't get to your questions in the meantime, we will be looking at getting a response to you over time. So now I will ask Brian to introduce our next speaker.
Steve not there? Is Steve not there? Hello?
Yeah, I'm here, Brian.
Oh, away you go.
Oh, you want me to do it? Okay. G'day, everyone. I'm Steve Abuscular, the Vice President for the Society. What a great initiative we've got going on here, and thanks very much to our President, Julie. I'm delighted to introduce our next speaker, Bethany Norstorm. I hope I said that correctly. She's from the University of Western Australia and will be speaking about the assisted colonisation of Western swamp turtles. Wow, that's a mouthful. Over to you, Bethany.
Thank you. I'll just set up my sharing my screen. Oh. Is that showing the presentation or still just the PowerPoint version?
The PowerPoint version.
Okay, just let me, I'll just have to switch that to the other screen, I think. There we go. I think that should be better.
Great.
Okay, thank you. All right. So thanks for having me today. As said, I'm going to be speaking about my PhD research, which is on the assisted colonization of the Western swamp turtle. So the critically endangered Western swamp turtle, or tortoise, is considered one of Australia's rarest reptiles. The swampy is a very small freshwater turtle, which is endemic to Southwest Australia. They're slow to grow and they only reach sexual maturity between the ages of 12 and 15 years. And they only lay one clutch of eggs per year with about 3 to 5 eggs. The swamp turtles rely on seasonal clay-based swamps. And very interestingly, the turtles are active in the winter wet months where they feed and then reproduce. And then they estivate throughout the summer, the hot summer and autumn months. However, these seasonal clay-based wetlands are in a bit of trouble. So they used to extend all along the Swan Coastal Plain in Western Australia, but this area is also the most densely human populated area in Western Australia. So the region has actually been extensively cleared for development and agriculture, and it's estimated that about 90% of these seasonal clay-based swamps have been cleared or drained. So there's only one last wild self-sustaining population of Western swamp tortoises, and it's restricted to one small fragmented and isolated reserve just north of Perth in Ellenbrook Nature Reserve, which is an area of about 80 hectares and contains about 40 mature adult Western swamp turtles. The turtles are also threatened by climate change. So winter rainfall in Southwest Australia has declined by more than 20% over the last 50 years, and it's projected to decline further. So this results in shorter hydro periods in seasonal wetland ecosystems, and therefore shorter activity periods for the turtles. If critical periods become too short, they won't be able to meet energy requirements needed for reproduction and growth. So just a bit on Western conservation work that's been done. By the 1980s, there are actually fewer than 30 turtles left in the wild. And there was a captive breeding program that wasn't doing very well. So Dr. Gerald Kuchling came over in 1988 from Austria and revived that program. And in 1991, he was able to hand that over to Perth Zoo. And it's actually been so successful that this breeding program has been able to release over 800 captive bred turtles to different nature reserves just north of Perth. However, there is uncertainty about the habitat suitability in these nature reserves due to climate change. So the big question now is, what can we do in the face of a drying southwestern Australian climate? And one option for that is assisted colonization or assisted migration, which is the intentional translocation of a species outside of their historic range to mitigate a threat, such as exotic species, disease, human impacts, or climate change. And assisted colonization is being considered for the Western swamp turtle as they face cumulative threats. So they've got that habitat loss and the climate change, which has resulted in them losing their ability to shift their distributions naturally. And then if we add their low reproductive rates and long generation times, it makes them even more susceptible to extinction. So with this assisted colonization, the Western swamp tortoise is actually the first species in the world to undergo it as a way to mitigate climate change. Previous research has resulted in the selection of assisted colonization sites a few hundred kilometers south of their current range in East Augusta and Murup. These areas have lower water temperatures and they have slightly longer hydro periods than in their natural range. And 2 short experimental assisted colonization trials have occurred starting in 2016, and the East Augusta site is looking quite promising so far. So that's where my research comes in. I'm building off previous assisted colonization trials to understand whether southern translocations will be successful. And I'm doing that by exploring 3 main aims. So the first is to conduct another trial, which actually started in August of this year. So I'm based in the field right now, actually, and I'm monitoring turtle behavior, microhabitat use, growth, and prey availability of about 75 turtles that we've released. So the turtles are all fitted with these tiny little radio trackers that also have built-in activity and temperature sensors, which will help us understand what the turtles are doing and when they're capable of feeding. So these tags transmit to a stationary receiver set up in the wetland, which collects the data, and then I can download on a weekly basis. The second idea of my PhD is to develop environmental DNA methods. So the basic idea of environmental DNA is that an organism interacts with its environment, and then DNA material is left behind through excretion or shedding, and then that genetic material can be collected and then identified. So I'll be developing eDNA primers to detect turtles in these southern assisted colonization sites. So as you can see from these videos, the turtles are extremely difficult to locate in the wild due to their small size, elusive behavior, and cryptic appearance. They really blend in their surroundings. And it's also very important to understand what the turtles are eating in their new environments and how they fit into the local food web. So I'll be able to look at this by using non-invasive DNA metabarcoding. So I can collect their fecal samples and then send them away for sequencing. And then from that, I can determine the composition of the prey consumed. And finally, I'll incorporate the results of this trial into predictive models, such as a dynamic energy budget model, which will allow me to quantify energy assimilation and usage throughout the turtle's life cycle. So this will allow us to understand things like reproductive age and rates, survival and longevity in these cooler southern sites, and whether they'll be ideal in the future. So just to sum up, I just want to say that this method of assisted colonization is not just limited to Western swamp turtles. As the climate continues to change, several species of multiple taxa won't be able to adapt in situ or migrate on their own. So this work will present a useful case study moving forward when attempting to mitigate threats of climate change using assisted colonization. And I'd just like to thank all the generous contributions and help I've had thus far. And finally, just mention the Friends of the Western Swamp Tortoise Group. They're a local community group here in Perth, and they've generously supported my project, and they support all Western Swamp Turtle conservation work. So if you're interested in joining the group, please visit either their website or their Facebook page, and they're always happy to have new members. Thank you.
Fabulous. Thank you, Bethany, for sharing that. Brian, would you like to introduce our next speaker?
Hi all, my name's Brian Scarsbrick and I'm also the Vice President of the Society. I'm delighted to introduce our next keynote speaker, Paul Wittine. Paul is the Chairman of the Friends of the Western Ground Parrot. But firstly, we will be shown a documentary on the Western Ground Parrot project, and then Paul will follow with a short summary. Thank you, Paul.
The western ground parrot of Western Australia is critically endangered. There are no more than 150 left in the wild, with numbers declining due to feral cats and foxes. Video journalist Mark Bennett joined conservationists. The western ground parrot of Western Australia is critically endangered. There are no more than 150 left in the wild, with numbers declining due to feral cats and foxes. Video journalist Mark Bennett joined conservationists on a bold but potentially risky operation to ensure a future for the enigmatic birds.
At a national park on Western Australia's remote south coast. The temporary headquarters for a team of scientists and volunteers on a mission to save Western Australia's rarest bird. The project-based camp at Cape Arid National Park is 10 hours drive southeast of Perth. It will be their home for 3 weeks. The project is the culmination of years of research, planning and preparation.
Bristol crew just need to go out with one of us tonight just to recalibrate because we need to be able to...
Another milestone on a long journey to save the critically endangered western ground parrot species. They used to be found around the southern Western Australian coastline. Vulnerable to wildfires, foxes and cats, their numbers are now estimated to be just 140.
Tumbuck track people need to leave at 45.
Without help, wildlife ecologists judge the bird is soon headed for extinction.
You know, it's been in the last 20 odd years that they've disappeared from, 2 of the 3 areas that we knew they occurred in the late 90s. That's very dramatic and, quite likely a result of the fact that they are a ground-dwelling parrot and really susceptible to threats of feral cats and foxes and fire regimes.
The aim is to establish a second population of the birds by relocating a few captured here
Sarah and Finn
to an area where they used to live, further west along the south coast.
In the last 5 years, we've had over 80% of the habitat in this reserve impacted by bushfires. And being a ground-dwelling parrot, having dense vegetation to roost and nest in is actually critically important. And so those areas have actually become quite limited in this environment.
It's the first attempt to move them to a new habitat. There's been some concern it could put at risk an already depleted population. But it's also a risk to do nothing.
So fingers crossed, we've all got everything crossed that this is going to be a success. We've been doing a lot of work here and in the proposed release site to try and make sure it's as safe as possible for these birds.
Yeah, you can just forget about the top one.
Along with the professionals, are biology master's graduates from the UK, now on an exchange project with the University of Western Australia.
The ground parrot is endangered, and to be involved in such an important project and to be able to get up close with such an important enigmatic species is just so incredible to just be a part of it.
Research and preparation for the translocation has been more than 5 years in the making, involving conservation professionals and volunteers from around Australia and New Zealand.
This setup here is our emergency care setup. We're hopeful that we won't need to use it. However, when we're working in such a remote location, it's very important that we've got options available. These birds are very rare and very special. So we have a dedicated aviary complex at the zoo where we house a very small number of western ground parrots. You know, the zoo has a lot of husbandry experience and veterinary expertise that we can contribute to this program to set things up for success, and we're hoping things go smoothly and yeah, we've got the best people involved.
But the bird's secretive habits don't make that an easy task. The western ground parrot is wary and well camouflaged, rarely heard and difficult to find or even see in the wild. And they're even more difficult to capture.
The plan is we're going to listen here, which looks like really good habitat for ground parrots.
Sarah Comer's team has spent many years analysing the location of bird calls in the park, recorded by remote microphones. To pinpoint their target, teams of listeners go out before dusk and dawn to just stand for hours and listen silently for its unique song. In the vast landscape covering hundreds of thousands of hectares and home to more than 160 species of birds, it's a veritable needle in a haystack. After a week of listening, this area is chosen to set the mist net traps.
Yeah, we've set up a bunch of mist nets behind us. There's about 280 metres of mist net with the hope of catching them tonight.
So this area's been identified as being potentially a good one because of the listening that's gone on over the past few days?
Yeah, so the data from our ARU analysis and having people on the ground listening has shown that the birds are moving from this way where they're feeding into their roosting sites.
So this is a very critical moment for when the sun has just gone down. They have about an hour window of opportunity where the birds leave where they've been feeding for the day and flying back to their roost, and hopefully through these mist nets. After months and years of preparation, the sound these researchers have been waiting to hear. Over the next few days, 6 more birds are caught.
Nice to have another one down.
Fantastic.
Good work.
But before they are relocated, each bird's health is checked. A Perth Zoo vet takes samples, photographs,
which is good. It doesn't seem to have feather loss.
And measurements.
It looks good.
So researchers can track the birds at their new home. They're fitted with a harness holding miniature transmitters and released in their new territory. This is the area where the 7 ground parrots have been brought from Cape Arid, 600 kilometres to the east, and relocated to this area where 18 years ago ground parrots thrived before foxes and cats decimated the population. So wildlife experts have extensively baited and trapped the ferals in a bid to give the new arrivals a fighting chance. It's now a few weeks since the release, and the faint pulse coming to the radio tracker transmitter is pure music to Sarah Comer.
How cool is that? This is the 3rd female that we released, and she's still fairly close to the area that we let her go in. Yeah, she's somewhere in here.
So far, the signs are good.
Or they all travelled well, they've all come out of the box really well, they've been moving around with their transmitters. That initial 7 days was our main period of concern, so the fact that we're working in an area that's unfenced. We've been doing a lot of introduced predator work, but you never know whether that's enough. So the fact that they've survived, they're obviously finding enough food to eat, and they've been able to move reasonable distances. We're feeling optimistic at this point.
But it's just the beginning. The next hurdle, securing ongoing government funding to control the foxes and cats, and possibly more relocation attempts in the future.
Well, we'll certainly be looking at how successful this program is. I mean, to date, it looks like it has been successful. We've successfully moved a number of birds and we'll continue to track them. And if further funding is required to support this population of parrots, I'll certainly be happy to advocate for that.
A tiny, enigmatic bird that has become a symbol and a focus for saving threatened Australian wildlife.
The ground parrot's a bit of a flagship for good conservation management, but there are a lot of other species here that will undoubtedly benefit from the work that we're doing too. But I think, we need to, look at restoring our native species into landscapes and building, populations that are resilient to the bigger picture things like climate change.
A mission and vision that resonates with a generation worried about the planet their children will inherit.
I feel like our generation, it's either going to be make or break, like it's either going to disappear in our generation or we're going to manage to turn it around. So if in any way I can help to turn it around, then that's great, but I think it's going to need a lot of effort from a lot of people.
With conservation, you have to be optimistic. There's a lot of challenges that our natural world faces these days, from climate change, and a lot of species are threatened with extinction. So if we're to turn things around, you have to be optimistic and maintain a positive outlook.
So it was a great introduction to the project there. It looks like we may have lost Paul from our panelist list. So what we might do just for now is move on, but if you do have any questions, we will be sure to answer them from here on. So what we might do is introduce the next speaker, and that is Quail.
Hello, everyone. My name is Wayne Greenwood. I'm a director on the board of the Society, and it is my pleasure to introduce our next speaker, Shay Jones, from the University of Wollongong, who will help us understand if Abuscular, Mycorrhizal fungi help grasses in heat wave. Do I get a plus for that, Shay? Did I say it correctly?
You're pretty close. So it's Abuscular Mycorrhizal fungi. But it's a bit of a mouthful, so.
Right. Off you go, Shay.
Yeah, I'll just try and share my screen. Hopefully that's working. Are you guys seeing my screen or?
No.
No.
Okay. Don't know why it does this. Oh, Lord.
That's
Sorry.
okay. No stress.
Shay,
take your time.
There's a little blue button. When you click share screen and you select the screen, down the right bottom corner, it says share. That's what shares it.
Yeah, so I'm clicking it and it's not showing up in the, it's just not loading.
Rats.
So another thing we can do, okay, so Paul has just got back and saying that his Wi-Fi cut off, but what we can do is either you can send the PowerPoint to me and I can share it from my end, or we can move on and then you can come back when you're ready and loaded. How are you going there?
Yeah, maybe we'll try and come back and get back to it when I'm loaded. I think I've just got to change some settings on my browser, so hopefully that will work.
Okay, no worries. Well, then we might move on to our next speaker. And again, Wayne, would you like to do the introduction?
Okay. Fast and the Furious here. It's my pleasure to introduce our next keynote speaker, Tamiel Brunt from the University of Queensland, who will be speaking about platypus conservation and what you can do to help preserve the platypus. Thank you, Tamiel.
Wonderful, thank you for having me. Can you see my screen?
Yes.
Wonderful, great, we'll get into it. Let me just... So thank you for having me. I would first like to acknowledge the First Nations people as the traditional owners of the lands on which my research was conducted. And I recognise country being home to many groups that I've been across southeast Queensland. I pay my deep respects to elders past, present and future. One of the local platypus names is Wudgeon from Yugambeh language, and he's the guardian of Scrubby Creek. I encourage you to go and Google Wudgeon and the beautiful story that he tells of protecting Scrubby Creek here in southeast Queensland. There's really is nothing quite like seeing a platypus in the wild. It really is just mesmerising, and you can waste hours captivated by their swimming. I was lucky enough to see a platypus in the wild at Warrawong Sanctuary in South Australia when I was completing Year 10 work placement. It's not so much the wild, it is an enclosed man-made billabong system for platypus, but it was still in a bush setting, so it was good enough for me to say the wild. But my recent journey started in 2014, and I volunteered with CSER Australia on a 3-day trip to the Grampians to help survey platypus in the Mackenzie River. We caught 3 platypuses in that trip, and it really was amazing to be up close and personal to such an iconic Australian species. And for me, I had a moment with the little female platypus pictured. I really connected with them during that trip. I learned so much during that time from ecologist Josh Griffiths, and their plight in Victoria just blew my mind. And because they are such a cryptic species and not readily seen, they are just easily forgotten about. They are a species that will disappear and we'd be none the wiser. Surely the same thing was happening in Queensland, and that really scared me. So I came back to Queensland ready to jump on board any research that was happening with this species. But unfortunately, there wasn't a lot going on. Compared to other states that have 20 and 40 years of ongoing research, Queensland was very sporadic with projects, and there wasn't a lot of longitudinal data, especially in areas where they were highly impacted by us. With the species being upgraded to near threatened on the IUCN Red List due to localised declines in these study areas that were found, I had no doubt that Queensland platypus were probably at risk, and we were just lacking the data to prove it. Throughout my Google search, I found Wildlife Queensland Platypus Watch Network, and they committed to observational surveys and community engagement. I eagerly contacted them and was lucky enough to develop my honours project, researching platypus requirements and diet assemblage in a local population west of Brisbane. I finished my honours and realised there was really a lot more to do with Queensland platypus, to raise their profile, to try and build up that data we were so lacking. So I continued my journey into my PhD project. In 2016, alongside Platypus Watch, we wanted to understand what the current distribution of platypuses were in our Brisbane waterways, and to get on board with a fairly new technique called environmental DNA, which Bethany touched on in her talk. Because we looked at historical records across the whole state of Queensland, we saw that there was very much a decline in those records. And did this mean that people were losing interest and not recording their sightings that were really important for us? Or were we actually losing platypus? Either way, we were quite concerned and wanted to understand what was happening. So we developed an eDNA project based upon historical sightings and chose multiple sites along waterways within Brisbane. This method revolutionised the way we can monitor the cryptic species. Just by taking the water sample, we can detect their presence. As Bethany said, because species poo in the water, they shed their hair cells and skid cells, we can pick up the small fragments of DNA through a 0.22 micron filter. We would pass up to 500 mils of water through the filters, and that became the sample. We would send these filters to the lab, which is EnviroDNA down in Melbourne, and they would run the PCR to amplify the specific platypus primers that would say detecting platypus presence or absence within our samples. It's 95% sensitive to be able to pick up species, so it certainly is amazing for these cryptic animals. But unfortunately, at the moment, it can't tell us number of individuals, relatedness, or precise locations. But hopefully our technology will advance far enough that we can certainly do that. We have completed sampling across the Greater Brisbane, and we have just finished our 6th year of the program this year. We've sampled over 200 sites in 71 waterways and detected platypus presence in 25 of those. In 21 of the historical waterways, we repeated sampling at least 2 to 5 times over these 6 years. And unfortunately, we can no longer detect platypus in these historical areas, such as Kedron Brook, which is just north of Brisbane. They have literally disappeared right under our noses within the last 16 years. We've also been able to use this eDNA data to develop occupancy modelling to determine habitat and environmental variables that influence platypus, specifically in an urban setting, because there is so much compounding on our freshwater waterways. The highest ranked model that you can see for platypus occurrence included slightly positive effects for our coarse organic matter and overhanging vegetation, but the topographic wetness index was certainly strong, and that relates to water, which is obviously highly important for platypus, and also the complex benthic substrate, which is the cobbled substrate that they feed through and is obviously really important for their food sources. And so these factors relate to the food, the shelter, and mating resources for platypus, which is obviously very important for them to continue persisting in urban areas. So this information can be used to guide management plans for the survival of platypus within our area. From this data and other records, because now I knew where platypus were, which is great, it's a great advantage, I was able to further develop a capture and mark survey as part of my PhD. So I've been capturing platypus to gain further genetic information about individual populations in hope to determine areas under threat from isolation and therefore the restricted gene flow. I used Fike nets, which are these D-framed nets that have a windsock out the back, and they have wings coming out the side. So we're blocking off the creek, one facing upstream, one facing downstream, in hope that platypus would swim into that windsock. It's half in and out of the water, so they can swim in, but obviously they can still come up to breathe and be protected. We would place a few of these sites throughout the creeks in the afternoon, and then we'd be checking them throughout the night every 2 to 3 hours for platypus, fingers crossed, or any of the bycatch that we might capture, like eels, turtles, or catfish. When I was lucky enough to capture a platypus, I would take some measurements. Firstly, I would grab it by the tail out of the net, because obviously the males have a venomous spur, and I definitely don't want to be tickled by that. And they would be placed into a calico bag and then onto a foam pad, and I would take some measurements such as the length, the weight, some morphological measurements of their bill. We can understand their body condition by how fat their tail is, how much fat they store in their tail. And so by like squishing their tail to see if it is quite rigid or it folds, we can score their body condition. Obviously, we can sex them due to the presence of the male's spur. It also gives us a bit of an age class as well, because as a male platypus ages, that spur sheath that is quite thick will wear down over time to a very impressive hypodermic needle. I would take a small snip of DNA from their hind foot webbing, and I would also microchip them for identification if I was to recapture them. This was one of my handsome platypuses I caught in a waterway in Ipswich, just west of Brisbane, aptly named Mr. Darcy, if you know Jane Austen, and a very swagger male that he was. And so overall, this information will hopefully feed into local policies to help raise the profile and protection of this species. Specifically, I'd really like to contribute to the national effort that's happening with quite a few organisations and the University of New South Wales to upgrade platypus to threatened on our EPBC Act. But we are having a lot of impact within our local area with councils, and they're getting on board to make platypus a priority species within their local government areas. There is a long way to go in Queensland, but the silver lining is that, thank goodness, they are a pretty robust little creature. But we are building up our community and awareness and expanding our projects and making that connection that's really important. This allows us to step up and make the changes necessary to protect this species now before it is too late. And you can help at home by taking some small steps to reduce the impacts on our waterways. Please scan the screen so you can download the information sheets, or it can be circulated afterwards. We impact the natural world around us every day, so it really is now our job to step up and make species like the platypus survive into the future. Our connection with nature has to be strong, not only because platypus need it, but we heavily rely upon it as well. Thank you.
Fabulous. Thank you so much, Tamiel. Just one question about specifically relocating platypus into areas where they previously occurred. Is that something that is that might happen in the future?
And look, I think, you know, there's always been very much a taboo around relocating species, but unfortunately we are impacting at a fast rate, and we're going to have to think about these kind of methods to help gene flow and gene recovery in populations. And I know that the New South Wales group, I think, is looking into repopulating a national park close to Sydney. I'm not too sure of the time frame on that, but you know, it is something that we need to talk about. We need to get serious with some of our species that, you know, we need to probably start saving in terms of that genetic recovery. So it's a lot of research to understand, you know, where are we going to get the individuals, what populations, where are we going to put them? But it's definitely something we really need to start talking about, and for it to not be a taboo topic anymore.
All right, excellent. Thank you so much again, Tamiel. And we will have our next speaker introduced by Phillip.
Thank you. I'll skip this. Hello, yes, my name is Phillip Sansom and I'm a director on the board of the Society. It's my pleasure to introduce to you our next speaker, Jenna Draper from the University of Adelaide, who will be speaking about the conservation utility of the Malay-rich flower shrub to arid zone frugivores and pollinators. Thank you, Jenna.
Alrighty, I'll get us started now. I hope you can all hear me.
Yep, you're all good.
Excellent. There we are. I think we're up and running now. So thank you for the introduction. So I'll be telling you about my AWS funded research on the conservation utility of Pioneer microcephala to arid zone pollinators and frugivores. So to get started, I'll tell you a little bit about what I mean by conservation utility. I'll unpack my title a little bit. So what I mean is I want to understand how useful Pioneer microcephala is for helping to conserve other species that are found in the arid zone. So what resources might it provide to arid zone species so that through conserving Pioneer microcephala, we can then conserve arid zone species too, maybe some of our more charismatic animals, for example. So the reason I chose Pioneer microcephala is because it's got a couple of different characteristics that make it sort of a really good candidate for further research into this area. So the first one that attracted my attention is that it's insect pollinated and it has these really sort of generalist pollinated flowers. They're really open and accessible. So hopefully a lot of different insect species access them for energy. Additionally, they produce these really, really bright, sort of juicy looking fleshy fruits. So I was also hoping that maybe I could understand whether or not there are reptile species or bird species that are consuming these in arid habitats to get a little bit of water during some of the drier months. And perhaps the most interesting characteristic of Pymele microcephala is that it's actually quite toxic. And it's been found to be very toxic to introduced livestock in particular. So it causes a number of nasty complications to cattle and sheep that may mean that it's actually graze resistant. So a lot of these animals won't eat it by accident. They accidentally get fed it into their feed. And then that's how we found out ultimately that it's toxic. So considering all of these characteristics, this made me wonder, so does this mean that Pymele microcephala could be a native specific chemically protected arid zone food resource? So to get to answering this question, I first needed to find out what arid zone fauna were consuming these floral resources and fruits, because I need to know whether or not the plant is toxic to them also. So I need to do some observational studies. So I looked at both pollinators and frugivores, which may have been reptiles or birds. I did this first using binoculars and sitting there and watching by eye to see if pollinators and frugivores would land and interact with the plant. But thanks to the AWS, I was also able to obtain a camera trap, which is triggered by time-lapse photography, so I can get some photographic evidence of various frugivores interacting with the plant. The study was conducted in South Australia in a nature reserve called Hiltaba, which is managed by the Nature Foundation and is found on the Eyre Peninsula. And also thanks to the AWS, I was able to get some money to get myself there, which was very important to access the very pristine open woodland habitat that they have there. So getting right into it, regarding the pollinators that I found on my trip to Hiltaba, it was unfortunately not very good weather for pollinator observation. It was very cold and it was very windy. So that made it very difficult for all of the little flying around pollinators to be able to be active during this time. So while I wasn't able to observe pollinators on this trip, it's still quite likely that a diversity of generalist insects are accessing Pymele microcephala flowers in the arid zone. And this is all based off of insects that I've been very lucky to observe in other sites. However, further observation will be needed to work out the exact species diversity that's found there. Regarding the frugivores, I got a lot more lucky when I was on my trip, and I conducted 25.75 hours of observation, with 17.5 of those hours being done by camera trap. And then the additional hours by eye were more so useful for observing the specific consumption behavior, actually watching the frugivores pluck the fruit off and throw it down their throat to make sure they're actually eating it. So during my trip, I observed 4 species consuming Pymele microcephala fruits. The first of these was the noisy miner, which we can see here sort of hopping around and plucking off all of the little fruits from the tree. By eye, and unfortunately not by camera trap, we also saw butcherbirds and wattlebirds consuming the fruits as well. And we even got very, very lucky to get some camera trap footage of the Australian ringneck sort of hopping around. We have here a breeding pair, which one of them will look out for the other while the other one goes and has a bit of a feed. And they were spending about 8 minutes stripping all of the fruits off of this plant so they can get some water and nutrients during the rather dry time of the year. Interestingly enough, the wattlebird that we observed consumed about 19 fruits in the space of about 30 seconds, which would almost certainly equate to what would be a lethal dose for humans. So it does seem like they can consume the fruit safely. So in conclusion, it looks like Pymele microcephala is a chemically protected native specific food source. My future avenues for this research will be confirming the pollinators to make sure that they actually are visiting the plants and finding out what species are visiting. And I'll also be expanding the frugivore study to additional sites. So thanks for listening. If you've got any questions that you don't pop in the chat, you can take a screen grab of this slide and you can email me at my address here below. Thanks.
Fabulous. Thank you so much, Jenna. And so we'll move on to the next speaker, and Ken will be introducing our next speaker. I'll just ask Ken to unmute.
There you are.
Okay. Thank you.
Yes, thank you. My name is Ken Mason, a director on the board of the Society. It's my pleasure to introduce our next keynote speaker, Kerry Allen from Regional Victorians, opposed to duck shooting, who will be speaking about the conservation of our precious water birds. One would wonder why anybody wouldn't be opposed to duck shooting. Well, there it is. Thank you, Kerry. Over to you.
Yep, you get that?
Thank you very much, Ken. I'm just trying to sort out the video now, actually. It's telling me the host has to start it.
Okay, no worries. I will go in and I've got that saved. So I will share my screen. Okay, can you see that?
I can.
All right, I will go into, all right, you just tell me when to go next.
All right, thank you. So Ken, thank you very much for the introduction, and thank you to Australian Wildlife Society for giving us the opportunity to take part in this. We're really pleased to be able to be here. My name is Kerry Allen. I'm spokesperson for the group Regional Victorians Opposed to Duck Shooting. We're a not-for-profit incorporated association based in Victoria, whose prime goal in life is to bring awareness to the public of what is actually happening at so many areas around the state, around the country, unfortunately, with our water birds, and to advocate for change. Now, the left-hand side of this first slide, I apologize, it's quite detailed, but we wanted to share with you a copy of a full-page ad that we ran in conjunction with some of our alliance partners, and Australian Wildlife Society is one of those. You'll see them listed in the third column on the right-hand side down the bottom there. So this was a full-page ad that we ran in The Age almost a year ago now, and it was an open letter that we wrote to the Victorian ministers, basically encapsulating the main points of why we believe bird shooting should be banned. Now, I know it's going to be impossible for you guys to read this, but I'll just cover off the top 2 points. The first one is cruelty. Well, duck shooting is cruel. There's no ifs or buts about it, unfortunately. And many people may not be aware of what happens when a shotgun fires a series of pellets. It's quite different to when a rifle fires a single bullet. When a shotgun fires pellets, there is, for want of a better word, a spray of ammunition that is dispelled out into an arc. And depending on the distance, it can be quite a wide arc. So when you're talking about a moving target that is sometimes quite a distance away, you can see the issues here. Ballistics experts say that at least 1 in 4 of the hundreds of thousands of water birds hit in duck shooting each year, they're not killed outright, they're wounded and they suffer horrific injuries. We've had reports of smashed bills, legs that have been blown off, wings that are shattered, internal organs are pierced. They're horrific injuries that these poor birds sustain. To Ken's point, it's ironic that this still continues in what should be called a modern era. If an abattoir had that rate of wounding, it'd be shut down and the owners jailed. But anyway, the next point is economics, which is quite close to our heart. There have been studies done which show that regional communities in particular would benefit from a ban on bird shooting. And I'll talk to you a little bit more about the other points. If we move on to the next slide, please, Megan. Thank you. So suffice to say, we've all had a taste now of what lockdown is like. It's not fun. And unfortunately, scientists are saying that 75% of new diseases are likely to be zoonotic. So that means really as a result of what humans are doing to their wildlife and ecosystems. So Australia, oops, I'll just get rid of that. Australia does not have the best track record when it comes to species loss, and shooting our native water birds is what we believe not a good addition to our already poor track record. The photo there on the right, that's a poor little freckled duck. which has been illegally shot. The freckled duck is Australia's rarest native duck. It's thought to be one of the world's rarest, but sadly it's collateral damage. So we might move on to slide 3, please, Megan. Thank you. So we talked a little bit about species loss. Well, this is the state of our water birds. It's not pretty. Now, the Centre for Ecosystem Science at University of New South Wales has been running aerial surveys for decades, and they've compiled the most robust long-term data set available on what is going on with our native water birds along East Australia. Now you can see there in the graph on the right-hand side, if this trajectory continues, you're quite possibly going to see some of our native water birds go extinct as well in the not too distant future. The data showed, so this is obviously on the back of what was in the news just recently about water bird numbers being 90% down since the 1980s, which is bad enough. Between 2019 and 2020, game birds, now remembering for most of the year they're protected, but when it's recreational shooting season, they get reclassed as game. 2019 to 2020, they fell a further 23%, and there was very little breeding despite the rain that we'd had. So that was a red flag. In Victoria, waterbird numbers fell 50% from 2019 to 2020. And the other stat that I wanted to mention was in New South Wales, waterbird numbers are half what they were just 5 years ago. It's heartbreaking. Next slide, please, Megan. Thank you. I'll just touch on this. The number of birds reported as killed is higher than what is published. Now, the reason we say that is because the data is based on a survey of shooters. Not all shooters are surveyed, so there's quite a room for error. Harvest estimates do not include that horrible stat that I was talking about before, you know, the 1 in 4 birds that are killed and fly, sorry, not killed and fly off wounded. So you can add another 25 to 67%, according to one study, on top of this data. Harvest estimates don't include the birds left behind, nor do they include the ripple effect through the species of shooting one of a monogamous pair. And a lot of our water birds are monogamous. Avian experts say that when we shoot one of them, it's quite possible that the surviving partner will also not survive, let alone repartner. So that figure down in the red circle there, that is just for Victoria, which is frightening. Slide 5, please. What is the impact to threatened protected species? You know what? The most scary thing about all of this is nobody knows. The Arthur Rylah Institute in a recent report actually said that that is a separate question that can only be properly addressed by gathering robust data on the rates of non-target species being killed or injured. Such data does not exist and would be extremely difficult to gather. Now, that was just horrific reading. And if you go to slide 6, please, Megan, what we do know is that as a result of documents we obtained through Freedom of Information, a typical landscape of collateral damage of our threatened and protected species is this. Now, this is just 1 year's worth of data. Now, the authorities seem to be a lot more robust and scientific years ago when they were collecting this data. Unfortunately, it hasn't been maintained. But this just gives you an idea of the types of species that are caught in the crossfire. You've got a barn owl, magpies, pelicans, swans. It's just heartbreaking. And unfortunately, this is the tip of the iceberg because this has only come from a small number of waterways which happen to be monitored. Slide 7, please, Megan. Thank you. Some other factors that are often forgotten about when we talk about bird shooting seasons, the impact to migratory birds is horrendous. Migratory shorebirds, for example, are particularly dependent on safe, peaceful havens at our wetlands to be able to rest up and feed up before they take off on their epic journeys up north to the Arctic, Siberia, Alaska. Now, unfortunately, disturbance does impact them. There've been studies done on that. But the other thing that is rarely taken into account is that often some species of migratory birds, first-year birds, don't leave the country, they stay here. So they are literally sitting in the line of fire for when recreational bird shooting starts up. Toxic lead. Lead has been banned in fuel and in paint for a reason. It's been banned in duck shooting in most places for the same reason, but illogically, it is still able to be used for other sorts of hunting. Lead does not break down. It stays in the environment for decades, if not centuries. And, we've probably got tons of toxic lead still sitting in our wetland sediments around the country, which is being picked up by water birds that mistake it for food. Pacific black ducks, I think, do this, blue-billed ducks. It only takes one lead pellet to cause organ failure and death. And it's a shocking, insidious death these poor things suffer. But then it's not just the birds that are impacted, it's secondary predators that feed on those birds, like eagles, as you can see in the picture here. Or is it a hawk? I'm not sure. White-bellied sea eagles, a lot of these birds are protected, secondary predators that are getting impacted by the lead in the environment. And lead, for some bizarre reason, is still allowed to shoot quail. So I shouldn't laugh, it's not funny, but we estimated that up to 5 tons of lead ammunition could be being pumped into just Victorian fields each quail shooting season. And some of those fields are used for food growing, mind you, for people. So we'll just move on to the next slide, please, Megan. Thank you. So on the topic of quail, it's not just about ducks. Poor old quail haven't had a lot of support. We've sort of picked up their cause as well because of what we found out through freedom of information, but there's very little, if any, data kept on quail populations. South Australia banned quail shooting season for the 2nd year in a row this year because of that. Victoria still had a full quail shooting season, 20 birds a day per shooter for the entire season. We're not sure why. The added problems with quail shooting are that quail closely resemble other species, for example, the critically endangered plains wanderer, and there's no accuracy test required of quail shooters. So we'll just move on. I think we're up to the last slide now. Or second last slide. Thank you, Megan. Just a heads up, what is at a very high level happening, that's it, around the country. So Queensland, WA, New South Wales have all banned recreational bird shooting. Now, New South Wales landowners can still get permits if they can prove that crop damage has been done by certain species. Now, you probably know that species like the pink-eared duck, for example, they don't eat rice. Their bills are not shaped that way. They actually eat the pests that eat the rice. So, they should be being protected, not shot. That's just one example. Restricted seasons, SA restricted duck shooting this year, banned quail shooting. Victoria restricted duck shooting, allowed a full quail shooting season. So that's the picture around the country at the moment. And then last slide. Hopefully, there we go. The issue with duck shooting is it also has an impact on a third group of stakeholders. So this debate, for want of a better word, is usually fought by shooters and animal rights groups. But what's been missing is the impact to regional people that are impacted. People that live and work with shooting happening in close proximity from before daylight for weeks on end. People that work night shift and they're trying to sleep during the day. People that work from home, which is becoming more popular. There's some horrendous adverse impacts to community from duck shooting. So we've run a couple of surveys on this, and some pretty frightening stats showed up. Over 80% of respondents had concerns for safety. Over 90% didn't believe that it benefited their communities at all. Over 25% reported that children were upset. Over 90% reported distress and anxiety. And yeah, we can relate to that. It happens not far from where I'm speaking to you right now. Over 30% reported adverse impact to stock. Horses have gone through fences. Over 30% reported issues with illegal camping, trespass, fires left unattended. That's a scary one because we know weather scientists are predicting fire seasons are going to become more severe and more intense. So we don't sort of want any unnecessary risks. Now we're moving to the last slide, please, Megan. This is it. So you can help. Anything from following us on Facebook, subscribing to our blogs on our website, volunteering. This is coming up to a really busy time for us. We welcome any time anybody has, they might be able to help in a small way, and make your vote count. It's interesting. We found some of our members even were telling us that they voted for their MPs, not realising that MP's stance on issues like this. So we think that maybe people sometimes vote habitually rather than really having an understanding of what the party's policies are on wildlife conservation, or even what their track record is on achieving wins for wildlife conservation. So we want to do a little bit more on that and help on the awareness side of things. So watch this space. We'll be bringing you some more information in the near future. So I've crammed a whole lot of information into a very short time. I'm sorry for rushing through. There's just so much to share. Please visit our website, reach out if you've got any questions, and if we've got time, I'm happy to try and answer some now.
Thank you, Kerry. We actually don't have any questions at the moment, but thank you to everyone who is submitting their questions through the Q&A box. So keep them coming, and no doubt Kerry will be able to answer any questions you may have through the Q&A. So we will now move on to our next speaker. And Hayley, would you like to introduce our next speaker there?
Thank you, Megan. Hello, my name is Hayley Starrett. I'm the director on the board, and it is my pleasure to introduce you to Gracie Liu from the University of New South Wales, who will be speaking to us about how we can improve frog conservation in fragmented landscapes. Thank you, Gracie.
We can't hear you. You're a bit muffled. Well, we can see the presentation. There's just no audio. No, I don't think that's going to work. Is anyone else having difficulty hearing Gracie? from the director specifically?
Yes.
Yeah,
Can't
okay.
hear. Just sounds very muffled.
Yeah, no, it's a complete blur. Can't hear a word.
You might need to change your speaking device, Gracie. Can you do it straight through your computer as opposed to your headphones? Oh, no, it's really not good. Try bringing it up a little bit more to your mouth. Yeah, no, still not good. That's interesting. I don't know how to get around this one. Do the directors have any suggestions? Or maybe we can move on. And
the only thing I could suggest is maybe if Gracie signs out and signs back in, try that.
okay, we might need to do that. Because we are at 12:06, what we might do is move on to Morrigan. Is Morrigan there with us? Yes. Okay. So if that's okay, Gracie, are you happy to stick around and come back to us? Okay. So a thumbs up. Okay. All right. Thanks. Wayne, hold on. No, we've got Trevor. Sorry. Can you please introduce our next speaker? I'm just
unmute. Am I there now?
Yep, unmute. You are here.
Yeah, hi, I'm Trevor Evans from, and I'm a director at the Society as well. I'd like to introduce Morrigan Ganane today to talk about something very close to my heart, Eastern Qualls. Yeah, I've bred Eastern Qualls here at Secret Creek now for about 20 years, so I'm listening with great anticipation. Welcome.
Thank you, Trevor. I'll just get my screen up. So I'm Morrigan Ganane. I'm one of the wildlife keepers at Guano Wildlife Sanctuary. So we're based here in Tassie, and I'm just here to discuss the release of some Eastern Qualls into the Central Highlands of Tasmania in 2020, so last year. So Guano was established in 1979. We're located in Mole Creek, so a small town in northern Tasmania, and very much have a focus on Tasmanian native wildlife and the conservation of those species. So we're currently home to over 30 different species of native Tasmanian wildlife. We take part in the rescue and rehabilitation of injured and orphaned wildlife. And then we also have breeding programs for 6 different threatened species. So they include the spotted-tailed qualls, eastern barred bandicoots, eastern bettongs, and some other dasyurids. So we've been breeding dasyurids for 40 years now. We've just bred Guano's 21st generation of Tasmanian devils. So it's recognised as the longest breeding program of any Australian native under stable conditions. And then we also breed 2 other species of dasyurids, so the eastern quoll and the spotted-tailed quoll. So the eastern quoll is listed as endangered by the IUCN and under federal legislation. Unfortunately, when we look at the mainland populations, they declined rapidly in the early to mid 1900s, right through to extinction in the 1970s. The species has survived in Tasmania, though it's estimated that there have been declines greater than 50% from 2000 to 2010. So Bruny Island is recognised as the last stable population of eastern quolls, and that is largely due to their cat control measures that they have on place. So feral cats and also foxes are a huge problem to these little marsupials. So the Tasmanian Quoll Conservation Program was established about 13 years ago, and it's comprised of a group of institutions all across Australia that manage the breeding of both eastern and spotted-tailed quolls to establish a genetically viable captive population and also be able to provide direct support to wild populations within Tasmania. Tasmanian Land Conservancy is a not-for-profit organisation that protects rare and valuable ecosystems throughout Tasmania, and working with the Tasmanian Quail Conservation Program, WWF, UTAS and Dipipwi, TLC set up a research project at their Silver Plains property in the Central Highlands in 2020. So this is planned to be part of a long-term project with the goal to use targeted releases of captive bred individuals to increase populations throughout the state. So raise them up to a number where they're able to breed enough that population declines are slowed and then eventually reversed, and they can continue to increase. So 20 eastern quolls were bred in captivity by the Tasmanian Quail Conservation Program for released, and they were then utilised by Tasmanian Land Conservancy's research project. So along with Truwana Wildlife Sanctuary, sorry about that, along with Truwana Wildlife Sanctuary, eastern quolls were also bred at East Coast Nature World and Devil's Cradle, so both locations within Tassie, so local areas. In November of last year, 20 eastern quolls were released, so there were 10 male and 10 female. DNA samples were obtained from the quolls prior to their release, and then they were monitored via tail-worn VHS transmitters post-release. Across the first 2 months post-release, there were feeding stations positioned to monitor activity with camera trapping, and then also live trapping and re-releasing occurred to check body conditions. Then researchers and volunteers retrapped those eastern quolls in July this year for further monitoring. One of the captive bred quolls had successfully mated and was carrying 6 young, which you can see pictured here. This was really exciting for everyone involved in the project because it does mean it's quite promising long term, as the breeding of these released quolls is obviously really essential to increase the population and the success of this project long term. Trapping is planned to occur again in December, and the idea with that is to obtain genetic samples from the juveniles that were bred this year. So that will give insight into the breeding success of the released cohort and also determine the impact they are having on the genetics of the population. As part of the project, just want to thank everyone who was involved. So Tasmanian Quail Conservation Program and Tasmanian Land Conservancy were a large part of it. WWF Australia, so their rewilding unit, provided funding, and also volunteers and staff as part of that trapping. UTAS and Dipipwi were also involved, and Meander Valley Veterinary Services assisted with the DNA samples of those captive bred quolls prior to their release. Funding was supplied by the Tasmanian Land Conservancy and the Wet and Haul Environment Trust, and also by WWF Australia, as I mentioned. Thank you.
Fabulous. Thank you so much, Morrigan, and keep up the fabulous work. We don't have any questions in the Q&A at the moment, but just a reminder to pop your questions in the Q&A box, and Morrigan will no doubt be happy to answer any that come through.
Absolutely. Thank you for the opportunity.
Thanks so much, Morrigan. Okay, so Wayne, you've got the next introduction.
Yes, well, it says our final speaker for the afternoon, but I think we're going to go back and we've got two people who are going to sort of slip back in afterwards. But our final speaker scheduled for this afternoon is Carly Molius from the University of Tasmania, who will be speaking about assessing microplastic exposure through non-invasive examination of guano in resident Tasmanian shorebirds. So therefore, you're going through the droppings to find meaningful answers. Is that correct, Carly?
Yes, that's correct. I've just shared my screen. Can you see that?
Perfect.
Great. All right, so hi, thanks for that introduction, Wayne. First, I'd just like to touch on a bit of background on microplastics in the marine environment specifically. So I've just got to move this. So plastic is the most common debris found in all marine environments. It's all over the world at all depths, even in Antarctica. We know the horrible effects larger plastics is having on marine wildlife, for example, suffocation, starvation, entanglement, and often death. However, understanding how microplastics are affecting wildlife is a lot more complex, and there is a lot of research going into this space at the moment. The official size of microplastics is anything under 5 millimeters in diameter. However, the size ranges are really immense, and as larger macroplastics continuously break up in the environment from biological forces such as weathering and degradation, the vast majority of plastics that are out there in the environment are minuscule, down to nano sizes. So because of that, it's extremely difficult to detect such tiny particles when doing research on microplastics. And it requires expensive and advanced technology, and likely the amount of plastics that is in the environment is heavily underestimated. So why do we care about these invisible particles floating around in the environment? We might think, well, it's inevitable they're going to exist on Earth, maybe indefinitely, but there is growing evidence how microplastic ingestion is having negative impacts on wildlife. So far, mostly the lower trophic organisms, such as invertebrates, have been studied and have shown a myriad of health complications, including changes in feeding rates, growth rates, fertility, increased inflammation, and mortality. Sadly, we probably have to accept that plastics are now a part of ecosystems. So it's important we understand how they're ingested by animals, how much the animals are ingesting, and also how they're transferred throughout the food webs. So onto my study species, two resident shorebirds, the hooded plover and the Australian pied oyster catcher. They can both be considered what we call sentinel species for coastal environments. Like seabirds in marine environments, they're upper trophic predators, which means they're higher in the food chain, and therefore we can use them as bioindicators to assess how healthy an ecosystem is. Both these species feed predominantly on marine invertebrates found in the sediment. So quantifying microplastic exposure can not only help us to determine how much the birds are consuming, it can also give us some interesting insight as to how the plastics are being transferred from marine to terrestrial environments. So onto my aims of the project. Firstly, we're hoping to create a baseline data set for microplastic exposure in shorebirds in Australia. This hasn't been done before. And secondly, to assess how these plastics are transferred in coastal environments through comparisons between what's found in the bird's guano and what's found in the sediment, and also comparisons between the two species as they have different bill sizes and therefore different foraging strategies. And thirdly, we hope to do this with as little disturbance as possible to the birds. And a way to do this is by examining their guano. Currently, I'm in the fieldwork stage of my project, which involves me going out to beaches around Hobart. I watch the birds through a telescope and until one makes a precious deposit, and then I go and collect that with a metal spoon. So back in the labs, the samples will then undergo a digestion process using an enzyme called trypsin, which is an enzyme found naturally in our intestines with the purpose to break up proteins. So it's able to digest that guano, leaving only the plastics behind. And then the microplastics will then be counted and measured using flow cytometry, which is a brilliant instrument that works by detecting the fluorescence of plastics. As you can see here, the plastics are fluorescing a bright blue color. So we can easily count how many there are in each of the samples. The flow cytometry is especially handy as it has the ability to detect those tiny nano-sized particles I mentioned earlier, which are usually really difficult to analyze and often left out of these kinds of studies. And also, it's actually never been used to quantify microplastics in wildlife before. So if it's successful, it could be a really exciting tool, new conservation tool that we can begin to use in the field. So that's the summary of my project. How can we as individuals and citizens help wildlife ingest less plastic? I guess first and foremost, reducing our plastic, our single-use plastic as much as we can. It's not just some fad about those single-use items. They, such as food packaging and plastic bags, they are commonly found in the ocean. And as they fragment into smaller pieces, they can be consumed by all sorts of species, in the lower trophic organisms, and then they make their way up into higher trophic predators such as shorebirds. So avoiding these single-use plastics as much as possible, as well as joining your local beach cleanup, which can help reduce the plastic entering our waterways and oceans in the first place. And that's all. I'd like to acknowledge the AWS for funding. This funding has gone through to laboratory expenses, such as using this expensive technology like flow cytometry, as well as my fieldwork and my fellow Adrift Lab team. And I'd also like to pay respect to the traditional owners of the land, which the research is conducted on. Thank you.
Fabulous. Thank you so much, Carly. And yeah, we are very honoured to be funding such important research with all of our university recipients. So keep up the great work. Just trying to unshare. So we'll move on and we will attempt to see how Shay's presentation is coming along. Perfect. Okay, Shay.
Sorry, so hopefully it works this time.
Looking positive.
Everyone seeing that?
Yeah, we're just not seeing it in presentation view.
Ah.
Perfect.
Is that good now?
Excellent.
Perfect.
Thank you.
So sorry about the hassle there, guys, and thank you for everyone for sticking around. But I'm Shay Jones, and I'm in my first year of my PhD at the University of Wollongong. My project is looking at Abuscular mycorrhizal fungi and its role in helping native grasses and native grasslands surviving extreme climate stress such as heat waves. So what are Abuscular mycorrhizal fungi? So it's a bit of a mouthful, so we tend to just call it AMF, but what it is a symbiotic fungi which live in or around plant roots. And if you can see, this image here shows what they look like under a microscope. So they're microscopic fungi that colonize plant roots, and both the fungi and the plant receive benefits from this association, with the plant getting increased nutrient uptake, increased water uptake, and also a potential increase in tolerance to stressful events and stressful environments. And these fungi are really widespread, and they are particularly common in grasses and grasslands. So Australian grasslands are probably, as you've guessed, characterised by vegetation communities dominated by grasses, but they also have a diversity of other flora and are home to some of our most iconic flora. But they are considered to be one of our most threatened ecological communities, with less than 1% remaining unmodified or undisturbed. And this is largely due to agricultural use, urban development, but climate change is emerging as a prominent threat to our remaining grasslands. And generally, public concern tends to be focused on other vegetation communities like rainforests or bushlands, and so grasslands tend to be forgotten. So it's important that we start understanding their importance and advocating for their conservation. So climate change poses some unique challenges on grasslands because they don't have a high canopy cover they are more exposed to high heat, and they also tend to exist in regions that are already at the limits of high temperatures and low rainfall. So they're already in challenging environments, and climate change is just exacerbating this even further. And so this increases their risk of natural events such as fires, and it makes them very vulnerable to extreme climate events such as heat waves and drought. So heat waves and droughts are stressful events by themselves, but we tend to find that they don't occur just by themselves. They typically co-occur. So during a drought, you have a greater chance of a heat wave occurring and vice versa. So we actually see these events being very challenging for plants because of this co-occurrence. Typically during a heat wave on its own, a plant will be able to increase its water use to cool itself down. And obviously during a drought, a plant will adopt a water conserving strategy to get by with the low water availability. So obviously when these events co-occur, they're not able to adopt one or the other, and so they tend to be forced into using conflicting mechanisms. And this is where AMF comes in, because AMF can improve water uptake, and it may be able to increase plant water use efficiency. So AMF could greatly improve grass species survival in the face of these events. So my project aims to research just that, and it's aiming to understand how AMF helps grasses survive heat and water stress events. So to look at this, I'll be undertaking a number of different heat and water stress experiments using native grass species, and this will be both in the field and in the lab. So I'll grow these grasses with and without AMF to see how AMF is changing grass species responses to heat and water stress conditions. One of my main aims is to understand how AMF may modify plant water use and uptake, and one way to do this is through carbon isotope analysis, which simply put just allows you to see how efficiently plants are using water, and that's what I'll be using my AWS funding to go towards the lab costs of that. So overall, my project aims to develop some understanding around grasslands in the face of climate change, and understanding the role of AMF in hopefully improving grassland survival, and also using this knowledge to predict how grasslands may look in the future. And I'm hoping that the results of my project can have some pretty broad applications across management and policy development, as well as ecosystem restoration and even some agricultural applications. So I just want to say thank you to Friends of Grasslands and AWS for funding this project, and also my collaborators at the Botanic Gardens, the Royal Botanic Gardens of Sydney, UTS, and the University of Wollongong.
Excellent, thank you, Shay. Okay, so we will go to Gracie next and see how the sound is working there.
Fingers crossed. Can you hear me this time?
Perfect. Yes.
Okay, great. Sorry about that hiccup earlier. And thanks everyone for sticking around. Okay. And we can see that all right as well?
Yep.
Okay, great. So this talk is going to be about part of my PhD research, which is centered all around how we can help improve frog conservation in highly modified and fragmented landscapes. So it should come as no surprise that human-driven habitat loss and modification is occurring faster than ever before. Hopefully my slides will change. Oop.
Just saying the thinking button on our end. There could be a potential problem with connection there. So it looks like Gracie may have dropped off completely. Sorry guys, just bear with us for a second. We'll see if she rejoins, and then maybe she can send her presentation through to me if she's able to reconnect.
Yeah, internet speed varies, so.
I just want to thank everyone for joining today, and we've had some very great talks. So thank you for sharing your time with us to help convey the important message of preserving our precious wildlife for future generations, and some very interesting technologies that are happening in order to do that on many different scales across Australia, and in different species who are living in quite varied habitats. So thank you so much. We hope that it's inspired the viewers to, you know, implement some wildlife conservation action. And if you would like to do a little bit more to help conserve our native wildlife, the links to all the presenters of our talks today are on our website under the Australian Wildlife Week tab. Also, the University Research Grant Award winners, their articles are listed on the University Research Grant page of our website. So you can go onto the website and read a little bit more in depth of each of those recipients' research on our native wildlife. So I'm just looking here on the side again. Unfortunately, it doesn't look like Gracie has been able to reconnect. She's got some fabulous work on frogs, so it'd be really worthwhile going on our website to check out Gracie's research. We might wrap it up there, but I will ask.
Read the article. Read the article in the magazine.
Yeah, read the article in the magazine. And I will ask Julie now to wrap us up.
I'd just like to thank everybody for attending today. This is our first webinar for the Australian Wildlife Week as part of the Australian Wildlife Society. I found all the talks really great today. And even though we had a few technical difficulties, I'm sure that we'll be able to get Gracie's talk up online so that we can all see it. Megan's been behind the scenes and right in front of us doing so much work, and I'd like to really thank her for all the work that she's done. And thank you to all the directors for participating and introducing the speakers as well. It's really great to see the diverse amount of research that's been occurring and activities that are occurring across Australia in terms of wildlife conservation, meaning fauna and flora, of course.
Yes, brilliant.
So I'm going to clap.
Yeah, me too.
Thank you, everyone.
Fantastic. Megan will be putting up the recordings from today's session up on the website. And I also want to thank everybody watching on Facebook. And I'd like to encourage you, if you aren't a member of the Society, to join the Society to help us with researchers across Australia and other organisations to working towards wildlife conservation. There were quite a few questions in the Q&A. Some of them got answered, some of them didn't. So we'll be collating those, and we'll get those answers out to you as soon as we can. Of course, if there's anything that the Society can assist everybody with, let us know as well. But overall, I'd like to just thank everybody for participating today, and hope that you continue to work towards preserving Australia's flora and fauna, and keep in touch. Thank you, everybody.
Thank you, Julie. Thank you very much. Congratulations. Well done.
Well done, Julie and Megan.
Yeah.
Thank you.